Yes, we're going to get started. I will start with a little introduction, and then I will let Dr. Rowe take it over. So this is the first presentation of our 2023 ASA webinar series. So the ASA is the Archaeological Society of America, and we had this webinar series last year, and it was kind of successful. So we wanted to do it this year, and we have the honor as to have Dr. Rowe as a first speaker. So I'm going to give you a little bit of background concerning Dr. Rowe education and his research topic, even though most of you may already know him. Dr. Rowe was born in Louisiana, where he obtained a bachelor's degree with a minor in chemistry from Louisiana State University. He continued his graduate education at LSU, obtaining a master's degree in physiology with a minor in biochemistry. And then he got a PhD in entomology with a minor in nuclear science in 1981. He accepted the position as assistant professor in entomology at the North Carolina State University in 1984. And his laboratory has two major research goals. The first one is the study of vector arthropods, including tigers, ticks, field flies, sandflies, and mosquito. The second objective is the improvement of agricultural production for food and fibers. So Dr. Rowe's research approaches include RNA sequencing, genomics, microbiomics, biochemistry, chemistry, arthropod, biology, bioassay, and application in the material science. Dr. Rowe has published over 350 scientific papers. He produced several major textbooks, including the two-volume Biology of Ticks. He trained as a committee chair more than 50 graduate students, produced over 30 patents, and finally, he has taken several products to the market through university licensing agreements and 4 North Carolina State University startups, which are active today. So pretty impressive career. I'm done now, and I will let Dr. Rowe give his presentation. And the title of his talk today is Ticks Are Not Insects, How Do They See, Smell, and Reproduce? Dr. Rowe, the floor is yours.
Thank you for the kind introduction. Between 1981 and 1984, when I joined NC State, I was a postdoc at the University of California at Davis in the area of cellular molecular biology. Just so people didn't think I was unemployed for 3 years, I was fortunate to get a postdoc in Bruce Hammock's lab. Hey, I'd like to begin my presentation by acknowledging 2 other labs that's been involved in this work. One is the laboratory of Dr. Dan Sonnenschein at Old Dominion University. And if you ever want to do something with a tick, Dan will know how to do it. He's an amazing researcher with a great deal of experience with rearing and doing experimental procedures with ticks. And then the other person in the other lab I would like to acknowledge is Dr. Charles Apperson. It was his vision of bringing our 3 labs together and to work on ticks that allowed all the things that I'll be talking about today that we have accomplished and did not accomplish, as you will see in the next few minutes. I'd also want to acknowledge the many young scientists that worked in our labs over the years. And this is not the full list, but it's been an honor working with these young people. They're now, most of them, well into their careers and doing fantastic work. And the work that you'll be seeing, most of that is from this list of the folks that you see on the screen. Too many to really go through every name, but I'm really thankful for that. And then for those in the audience that might not be familiar with ticks, maybe you work on other acarines, but maybe it's not ticks. I'll give you a little background about ticks. I mean, I didn't start my career working on ticks. I worked on agricultural pests, so I had a lot to learn. But they're blood-feeding ectoparasites. They're roughly divided into two groups, hard ticks and soft ticks. Most of the work that you'll be seeing this afternoon is with a hard tick, the American dog tick, Dermacentor variabilis. People are interested in ticks because they vector microbes that cause human and animal diseases. And many people don't understand that in the United States, ticks surpass all other arthropods in the variety of infectious agents that they transmit to people and animals. And some of the biggies that you've probably heard about is Lyme disease, Rocky Mountain spotted fever, but there are other microbes that cause human and animal diseases, some of which are listed here. This is on the left a photograph of someone that's infected and has Rocky Mountain spotted fever. This is transmitted by the American dog tick, Dermacentor variabilis, and is the tick model that we do most of our research on. And there are thousands of cases of this annual. The biggie, though, is Lyme disease, transferred by the deer tick, Ixodes scapularis, and there are hundreds of thousands of cases of infection from this tick. So this is a problem that people have with ticks, and why we want to know more about tick physiology. You probably haven't heard about the tropical bot tick, but this tick has been migrating from Africa toward the United States, and it's now reached the Caribbean islands off the coast of the U.S. And this tick is classified by the U.S. government, by Homeland Security, as a high consequence threat to the United States homeland. That's because this tick carries a bacteria that causes heartwater disease. And this disease kills livestock and wildlife. And because the tick is so close to the U.S. now, you could even accidentally introduce this tick to the U.S., or you could do it on purpose just by simply going to the Virgin Islands, U.S. Virgin Islands, with your pet and bringing the tick back. But ticks overall are a problem in animal production because you get enough ticks on the animal, it reduces the animal weight, it reduces milk production, and it could cause abortions. But the main thing that people don't like about ticks is you just don't want to walk around with a tick hanging from your earlobe. It's just not socially something that's accepted. And but it happens, or it's on your neck, or it's some other place that we don't even want to talk about where it might be. So that's one problem with ticks. And the other is that once you remove the tick, it can itch so bad you scratch a hole in your skin. So they're just a problem. And that's the reason they're so much interested in the study of ticks. This slide here shows you the life cycle. It has 4 stages: the egg, a 6-legged larva, an 8-leg nymph, and an 8-leg adult. And to go from one stage to the other, except the egg stage, it requires that the tick take a blood meal. That blood meal could be on the same host, or it could be on a different host, depending on the species of tick. And because ticks develop this way, like insects develop in stages, because superficially they look a little bit like insects, it's easy to try to understand tick development, tick biology, based on what we already know about insects, in which there's been a huge amount of research effort in the study of insects. So this will be kind of a theme for my presentation about ticks. Ticks are not insects. So our work in our 3 labs is focused on the reproduction of ticks. And there are 3 strategies for tick reproduction. One is they mate anytime. The other is they mate before or during feeding. Or in the case of the American dog tick, they must feed on the host both at the same time, and this occurs before mating. And this next slide shows you the life cycle of the American dog tick, which will be the focus of my research, our research in the next few minutes. So I'll go over the details of this because it's really important for you to understand this, to understand the work that we've been doing. The males and females must find the host at the same time. In the case of our research, the host is a rabbit in Dan Sunnenshine's lab at Old Dominion University. And males and females go on the host at the same time. Then the males feed completely, and then the females only partially feed and then they stop. And then once that female is partially fed, she releases pheromone, a chemical that attracts the males. The males detach from the host. They seek out the female. The male inserts his mouth parts into the female genital tract, then produces a spermatophore, which is a packet of sperm. And then that spermatophore is then transferred to the genital tract of the female. And it's that transfer of that spermatophore and whatever's in that spermatophore that triggers a whole cascade of changes in that female. And you can see that in the next slide here. That female undergoes what Dan calls the big sip, where it takes in a large amount of blood, and you can see the size of that tick when it feeds to repletion. And it initiates the vitellogenesis, the making of yolk protein and the acquisition of that yolk protein by the eggs. And then that female will drop off from the host, detach and drop off from the host, lay a huge number of eggs, and then it dies. That's the reproductive life cycle that we've been studying for some time now. And I'll be addressing 4 questions in the presentation today. How do ticks regulate reproduction? How do ticks smell or taste? What is the role of chemoreception in blood feeding, reproduction, and repellency? And what other ways can ticks find their host other than chemoreception? So the first is, how do ticks regulate reproduction? And I'm going to break this down to 2 steps. Step 1 is the female and the male, but we're focusing in on the female mostly. How the female decides to blood feed. And what happens is the female finds the host, is on the surface of the host. There are cues from the host that stimulates, and the brain processes that information and tells the female to attach to the host. And most of our work is done on rabbits. And then this virgin female blood feeds to the part-fed condition. That blood feeding then stimulates the tissues in the body to synthesize and secrete a protein we call CP, or the common protein. It's a heme lipoglycoprotein, and it's just a way for storing all the things that female is going to need later in life when it's making eggs. And the level of that protein increases during this first step in the reproductive cycle. The next step in that cycle, step 2, is what we just discussed, where the male detaches, goes to the female, and eventually transfers that spermatophore into the female genital tract. That stimulates this feeding to repletion and the initiation of egg maturation. And we've been interested in trying to understand the endocrine regulation of that process. So this area highlighted here, and I'll be using this as I go through this diagram to show you the different areas of this physiological scheme here. So something is likely from the testes or the vas deferens, is transferred into the female genital tract by the male. We'll call it for now a male gonadotropin. That tells the brain of that part-fed virgin female that's now mated to blood feed to repletion. That female will take in a large amount of blood and become replete, like the pictures I showed you earlier. What is this male gonadotropin? Can you still see that okay?
Yep.
All right. So A paper was published in PNAS in 2004 that said there were two proteins, EF alpha and EF beta. It was these two proteins that were transferred in the spermatophore that initiated this big SIP. So we developed a hypothesis. This was in Amblyomma herbarium. We drew the hypothesis that this is the same thing that's happening in the American dog tick. So the hypothesis was that EF alpha and EF beta was transferred in the American dog tick, and it initiated this blood feeding to the replete condition. To test this hypothesis, I don't know why this thing is not responding. Got any suggestions, Grayson?
Yeah, it's loading. So it looks like it's loading
really
really
slow
much. There
for
you go.
some reason. It's loading real slow. So to answer this question of EF alpha and EF beta in the American dog tick, we developed a transcriptome to the male testes and accessory glands from unfed and fed adults of the American dog tick. And we also looked at genetic data that was available for Ixodes scapularis and Rhiplocephalus microplus. And we could not find EF beta in any of these three tick species, but we did find EF alpha. And when we did alignments, the overall similarity was 96% between these EF alphas from these four tick species. These were males, 21% identical. So we were able to find EF alpha. When we knocked down EF alpha in these males, we could do a 96% reduction in EF alpha by RNAi. These were in fully fed males. And when we gave these males to a virgin, part-fed female, the males successfully mated with those females. And even in the absence of the message for EF alpha, at least a 96% reduction in that message, those female reproduction in the big SIP occurred normally, which suggested that EF alpha was not functioning as the gonadotropin in the American dog tick. So Dan did a number of really cool experiments where he started injecting things. This shows Dan injecting a solution into the genital pore of Dermacentor variabilis. These were all part-fed females that were not mated. And this is sort of a summary of what we found. We found that EF alpha was present in the American dog tick male and other ticks, but not EF beta. A 96% knockdown of EF alpha at the message level in males had no impact on females. They just fed, once they mated, they fed normally to repletion, so it didn't block that process. Dan showed that male courtship alone did not initiate female reproduction. He injected different things to expand the volume of the female genital tract, like, for example, glass beads. This did not start reproduction. But he did find that aqueous extracts of the testes and the accessory glands, when injected into the female genital pore, initiated female reproduction. And this factor to date has not been identified. So we know there is a male gonadotropin. The evidence that we have doesn't suggest it's EF alpha, but we haven't identified that factor yet. The other question that we were interested in is juvenile hormone. And the reason why is this juvenile hormone, shows the structure of this hormone. In almost all insects that have been studied, juvenile hormone regulates female reproduction. There's some exceptions to that, and I'll talk about those in a minute. But juvenile hormone is a very interesting molecule because it's synthesized using these metabolic pathway for making steroids. But in insects, they've lost the ability to make steroids, and ticks have done the same thing. And they use that pathway now to make this hormone, juvenile hormone. And so it's an obvious thing to think about, that maybe it's juvenile hormone that regulates egg development in ticks. It regulates yolk synthesis, and it regulates reproduction in insects. So the question is, do ticks have JH? So there was a paper published in Science in 1979 that was very convincing, that said ticks used juvenile hormone, this was with Ornithodoros parkeri, to regulate egg development. And it was a solid study. And when other researchers around the world read this paper, they published papers showing juvenile hormone was the hormone that regulated egg development in ticks. And 20 or 30 papers were published preceding the work that we conducted. But when we looked at the research that was present at the time that we started our work, we realized no one ever looked to see whether ticks actually had JH. So we wrote and successfully obtained an NIH grant, and the objective was to determine, do ticks make juvenile hormone? Because if they don't make juvenile hormone, then they can't use juvenile hormone to regulate their reproductive process. It's just that simple. We had a postdoc, Paul Neese, that developed this radiometric assay. He looked at individual different tissues or organs in ticks. He looked at them in combination. This assay was sensitive down to the low femtogram range. That's 10 to the minus 15th of a gram. And we could not find any JH that were known to be produced by insects or the precursor to JH, which is methylphenidate in these studies. So this was very puzzling. So we used a different approach, a GC mass spec study, to see if we could find JH in ticks. The problem is you have to have a GC mass spectrometer. So we had to write a separate grant to get a GC mass spectrometer. Then we had to figure out how to use the darn thing so we could do the studies. And there's already an established method, GC mass spec, to identify JH in insects. And we basically modified that thing to work with ticks. And when we looked to see if ticks had juvenile hormone, we couldn't find any of the insect juvenile hormones in Dermacentum variabilis or in soft ticks for that matter. So the third thing that Paul did is he developed this thing called a Galaria bioassay. This assay uses an insect, a wax moth insect, and it has a bioassay, a biological response if you expose this insect to a juvenoid, some kind of juvenile hormone. So we extracted lipids from ticks and we applied it to this insect system, and we couldn't find anything in ticks at any point in development that would act like a juvenile hormone in this insect bioassay. So we concluded from these studies that ticks did not make JH or the precursor to JH, which is methylphenidate. So if this is the case, then how do ticks regulate their reproduction? Again, as I mentioned a little bit earlier, the regulation of female reproduction or egg development is simply about the synthesis of the yolk protein. And in the shaded area, you'll see this is a native page gel. This is the yolk protein shown here. This is the carrier protein below it. You can see that in unfed adult females, they don't make yolk protein. In a partially fed virgin female, they don't make yolk protein. It's only once that spermatophore is transferred that female becomes replete, you begin to see that protein in the blood. That protein eventually ends up in the egg. So it's just about how that protein is regulated. It's just really that simple. And we knew from working in mosquitoes that it's not juvenile hormone that regulates egg development, but it's an ecdysteroid, 20-hydroxyecdysone. So our hypothesis is that maybe it's 20-hydroxyecdysone that initiates egg development in Dermacentor variabilis. And there was already work in the lab prior to meeting Dan for the first time, where he looked at ecdysteroid levels during reproduction in ticks and found that ecdysteroid levels were high during when they were making eggs. So it was a reasonable thing to investigate. And he simply injected 20-hydroxyecdysone into ticks to see whether when he did that, whether we would get the production of this biosynthesis of yolk protein. And this is the proof of that. He found when you injected 20-hydroxyecdysone, and we did northern blots to see the presence or absence of the message, that the 20-hydroxyecdysone would appear in part-fed virgin females still attached to a rabbit when you injected physiological levels of 20-hydroxyecdysone into that tick. And when you just injected a solvent, nothing happened. You never saw the message. This injection did not cause the big sip, but it did cause the development of the yolk protein. When you look at the growth of the ovary for a female tick that's been fertilized by the male, it shows the growth of the weight of that ovary. And when you inject 20-hydroxyecdysone at physiological levels, the size of the ovary is almost the same, even though you don't get the big sip. Statistically, there was no difference between the weights of these ovaries. And then finally, if you actually look at the eggs and the ovary of an a part-fed virgin female, you see the ovary looks like this. And then when you inject 20-hydroxyecdysone into that part-fed virgin female, you get well-developed brown eggs. And the eggs are brown because the yolk protein carries heme that they obtain from the blood meal that they take. So there's already information in the literature that there was an ecdysteroid tropic hormone produced by the brain that initiated the synthesis of 20-hydroxyecdysone in the epidermis. So the picture became clear that the 20-hydroxyecdysone initiated the synthesis of yolk protein, and we can see the yolk protein and the message in these ticks that were injected with 20-hydroxyecdysone. In insects, for the yolk protein to go into the actual oocyte follicle, you have to have a receptor. It's called the yolk protein receptor. So this yolk protein receptor was sequenced, and it was very similar to the receptor that's found in insects. And when we knocked down that receptor so that the message was knocked down using RNAi, as shown here in this shaded area here, there was no yolk protein receptor in the RNAi injected versus the PBS phosphate buffer controls. And these females, even though they made yolk protein, the yolk never entered the egg. So that sort of completed our understanding of how egg maturation occurs in Dermacentor variabilis via the ecdysteroids. But still there's the question, is there anything relative to the JH pathway that's found in ticks? So I had a student in my laboratory is now Dr. Zhu that works for the USDA, and his PhD work was to try to study this question. And this slide here shows you the biosynthetic pathway in insects for the synthesis of juvenile hormone. And contains a mevalonate pathway and a JH branch, and the sterol branch is line drawn through it because in insects and ticks, they cannot synthesize steroids. You have to get that from their diet. So the question was, does this pathway exist in ticks? So we dissected the syn ganglion, and when you dissect the syn ganglion, this is essentially the entire nervous system, and it includes the stomatogastric system of ticks. We developed transcriptomes to Dermacentor variabilis and a soft tick, Ornithodoros. And then we had genetic information also from Ixodes scapularis. And we simply asked the question, is these enzymes that are found in insects, are there orthologs of those in these transcriptomes that could explain whether these pathways exist in ticks? And relative to the mevalonate pathway, we found clear orthologs with very low E values for every step in the mevalonate pathway in the 3 ticks that we looked at. In terms of the JH branch, the only thing we found was farnesyl oxidase, which is suggesting that ticks might make farnesyl. We didn't have good probes for the other 2 steps in that pathway that are in red. We did find methyltransferases, a lot of methyltransferases, but JZ was unable to find the JH hydrophobic pocket in those methyltransferases, which suggests that they don't bind juvenile hormone. And then the monooxygenase that adds the epoxide shown here to JH, that epoxidase was not present in our transcriptomes. So the overall conclusion from this work is that ticks do not make juvenile hormone. Ticks have the mevalonate pathway. They don't have the CYP15 JH epoxidase, but they do have a large number of P450s. They have an expansion of the methyltransferase family, but it doesn't appear to be able to bind JH based on our modeling. We found farnesyl oxidase, and we hypothesized that maybe ticks can synthesize farnesyl, but we haven't yet identified farnesyl in tick circulation. And basically, at this juncture, we don't understand what is the role of this pathway in female reproduction. In mosquitoes, juvenile hormone doesn't regulate egg production, but does regulate and control the behavior of the mosquito so that it blood feeds. Or maybe this pathway has that function. This is just not answered yet. But the other intriguing thing we found in these transcriptomes is all of the neuropeptides, all the neurohormones produced by the central nervous system that in insects regulates the molting process. Molting in insects is regulated by a number of different neuropeptide hormones that regulates all the behavior and physiological changes that must occur during a molting process. These are in adult ticks that don't molt. So this suggests, since they're present, although we don't fully understand their role, it suggests that they have a role in reproduction. One of my PhD students just successfully defended his PhD, Andrew Dami, and he's now found that when you knock down some of these neuropeptides, the ticks can't produce fully developed eggs. So it suggests that there's something about these molting neuropeptide hormones that's involved in the reproductive process. So the next thing is, how do ticks smell or taste? And this has been a really fun project. And as you know, ticks don't have antennae. I used to work on insects, not ticks. But they have something comparable to that, and that's the hallux organ. And it's at the tip of the front pair of legs, and they use that like an antenna. And this shows you a view from afar of what that hollow organ looks like at the end of the front pair of legs. This is an SEM on the left that shows you the pair of ocelli that's found on the tick for vision. And this is a area of the leg where the hallux, this is the hallux organ. It has two areas, an anterior pit, which has these cincilla that stick out. Then you had this, what's called a capsule area, which I'll talk about in more detail in the next few minutes. In this anterior pit, you have these cincilla. Some of these are have multiple openings into the cincilla, suggesting it might be olfactory. But in some cases, some of these have only a pore at the tip of the cincilla. And then we have this Bobby Mitchell, who was working in this project along with Ann Carr, another student in the laboratory. Also found a number of other neurosensory organs associated with the end of that leg. This capsule is very interesting because if you look inside of it, there are a number of cincilla that project into a opening inside. These are multi-pore cincilla, but the pores are blocked for some reason. And then there are these glands that we think secrete some kind of solution into this cavity. Then you got this weird shaped opening to the outside. What is that all about? Looks nothing like an insect antenna, for sure, right? And what is all this stuff there? What is it being used for? This is what it looks like in the Amblyora americanum. And then the morphology is quite different in Ixodes scapularis. And I'm going to explain the reason, what we think is the functional reason for these differences. So what is the mechanism of smell and or taste in the American dog tick? No one knew. No one had studied this before. So this was the task that we had before us. So we approached this by extracting the RNA from the end of the front pair of legs and then the end of the back pair of legs, understanding the back pair of legs doesn't have this sensory structure that we thought was involved in chemoreception. And then we made transcriptomes to the front legs and the hind legs, and then we subtracted those transcriptomes. And when we did this, we found out roughly 2,000 contigs that were exclusive to the front pair of legs and potentially to the hallux organ. Now, there's definitely problems with this work. And I'm going to talk about the issues with the study a little bit later. But this is what we had at the time to work with. And I'll tell you what we found. We used a variety of bioinformatics tools to ask what is the possible function of these 2,000 contigs that we had found that was exclusive to the front pair of legs. Just to remind you, and for those that are not familiar with how insects smell, you have this stimulus molecule, an odorant molecule that enters this syncytium lymph. There are odorant binding proteins that bind these odorant molecules because they're typically lipophilic, and this is an aqueous solution that transports to a receptor, an odorant receptor in the dendrite. When the two bind, an ion channel opens up, you get an action potential, and you get a sensation that is registered by the tick or insect brain in this particular case. And then there are enzymes in this syncytial lymph that break down those odorant molecules and resets the receptor. So that's what is known in insects. And so we would have to assume, I guess, at the beginning that maybe ticks do the same thing. We found no evidence in our transcriptomes that ticks have insect or vertebrate-like odorant binding proteins or insect pheromone binding proteins in those 2,000 contigs. There was no evidence of gustatory receptors exclusive in the tick front leg transcriptome. Now, at the time we did this work, 56 gustatory receptors have been identified in the genome of the deer tick, Ixodes scapularis. We found 3 contigs in our transcriptome, but they were present in both the front legs and the back legs that appeared to be gustatory receptors. It wasn't exclusive to the front pair of legs. This is what we found, and we found a lot of stuff, but this is sort of a summary of some of the highlights of what we found. We found a lipocalin that maybe is a binding protein, maybe not. We found a complete GPCR pathway that was exclusive to the front pair of legs. We found this thing called a beta-arrestin, which is a terminating protein that blocks this GPCR pathway. And then we found a host of xenobiotic metabolizing enzymes that might turn over odorant molecules as they enter some aspect of the hallux organ. So this slide here shows you what the insect system looks like. Odorant molecules, binding proteins, receptors associated with a gated channel. This is what the tick system looked like when we started our work, because we knew nothing about how ticks smell. We didn't find any insect-like binding proteins. We did find lipocalins. It could be novel binding proteins. Or maybe there are no odorant binding proteins used in ticks. Seems unlikely, but that's at least was a possibility that we had to mention at the time. We didn't find the typical receptors that we see in insects. We did find a GPCR associated with the front pair of legs and a complete signal transduction system, which included also a cyclic nucleotide gated ion channel and a beta-arrestin, which is used to shut this system down. The next subject is the role of chemoreception in blood feeding and reproduction and repellency. What other ways, and then the last thing I'll talk about is other ways in which ticks can find hosts other than by smell. If you look at this hallux organ right here, I begged Dan many times, Dan, you probably can remember this, to cut that darn hallux organ off and place that female on a rabbit and see if it'll blood feed. And I didn't
Still remember.
You remember that?
I sure do.
And you would say, well, they're not going to blood feed, Mike. But finally I pestered you enough that you did the experiment. And lo and behold, blood feeding was delayed as shown in this top graph here. But the females blood fed normally to port-fed condition without the hallux organ. When we looked at repellency without the hallux organ, we found that the hallux organ was needed for the detection of a repellent, and the repellent that we used was D. So just to summarize all of our work with chemoreception, we found gustatory receptors, but they were not exclusive to the hallux organ. We didn't find any odorant binding proteins or odorant receptors like what are in insects in the tick first legs. We found a GPCR pathway that was exclusive to the front pair of legs, a complete GPCR pathway. The pedipalps and not the hallux organ is needed for blood feeding. The hallux organ is the site for the detection of repellents like D. But there's a problem with this work, and I think the work needs to be redone in the future. It's difficult to extract the RNA from this part of the leg because of the heavy sclerotization. So it was a challenge to get good solid DNA. And I think we need to redo these hallux organ, and we need to add pedipalp transcriptomes in the future at different stages in development. And then at the time, we didn't have a genome of any type to help guide the assembly of the transcripts that we had. And that was a problem in assembling all of this transcriptal data. Then RNA-seq at the time was in its infancy. So we didn't really conduct a legitimate RNA-seq experiment, and that needs to be done at different times of development. And I think if that can be done again this way with the current state of the capability we have today, I think we can have a better understanding of how ticks smell and taste. And the final thing I want to talk about, and I'm running a little bit late, but I had a late start because the problem that we had at the start, but are there other ways that ticks can find their host other than chemoreception? Now, I want to go back to this diagram of this capsule area. What is this about? You've got these sensilla, multi-pore, but the pores are blocked, projecting into this cavity under the surface of this capsule, probably filled with fluid. What is this? What is this for? Is this chemoreception, or is it doing something else? We know ticks have eyes. They have a pair of ocelli. Then they have this capsule area. What is this capsule area? Is it involved in chemoreception, or maybe it has a different function? And then Bobby found all these other things around the Hallow's organ, which we didn't know exactly what these things were for. So we did some very simple-minded experiments. We took Dermacentor variabilis and we exposed it to visible light, and they respond. They can obviously see the visible light and they respond to it. We cover the eyes with black paint, and then they don't respond to the visible light. Makes sense. We cut off the Hallow's organ and we leave the eyes intact, and they respond to the light like you would expect. Okay, that's fine. But the interesting thing is that if we go to infrared light and we put this tick in the dark and we turn on an infrared light source, the ticks crawl to it very fast, amazingly fast. If we cover their eyes and we turn on the infrared light, they walk to the light very fast. If we cut off the Hallow's organs, they don't see the light at all, the infrared light at all. They don't know it's there, which suggests there's something about the end of that leg. Could be the Hallow's organ, could be another structure that detects infrared light, as if they're seeing infrared light with their front pair of legs. This is a video. Hopefully it'll play. For some reason, when this thing is not operating like it did before. And we just ran this test yesterday and it worked fine. Maybe it's low.
Do you think it is because you have a laser on? Maybe if you remove the laser.
Yeah, the laser, I think, has caused everything to kind of.
If you click right and you just go back to your regular mouse, pointer option.
Yeah. The problem is I've got multiple screens here, and the
Okay.
images and the things are that's don't worry about the video because we're running out of time.
Okay.
I'll just explain what happened. So, and this is the case when the pedipalp, when the Hallow's organ is intact. This tick will quickly move toward this infrared light. I have a video. Trust me, I have a video of this. It played fine yesterday. Maybe because we're recording, it's changed some. I don't know what's going on, but this Dermacentor variabilis quickly moves toward this light. Now you can't see this with your eye. This is total darkness. But we had a video camera that could detect infrared light, so we could record this. When you remove the Hallow's organ, this tick would move and go the opposite direction, or it could go anywhere because it could not detect the light. So it was clear that there was something at the end of the leg that resulted in the detection of infrared light. And if you look at the structure of Dermacentor variabilis, the capsule, it looks like this, and this tick can detect infrared light. If you look at Amblyomma americanum, the capsule looks like this. This tick is attracted to infrared light. If you look at the capsule area here of Ixodes scapularis, it looks very different. This tick is not attracted to infrared light. So it's something about, we think, the structure of that capsule that is responsible for infrared light detection. And Dr. Ann Carr has now published a paper since his work that shows the same thing. So in summary, ticks can detect infrared light. How they use this in a practical way, I think needs further study. How we can use this for tick control, we're all over this, and I don't have time to discuss it, but we think there's a lot of opportunities with that. I want to finish this presentation to remind you to remember the story about the tick JH and ticks are not insects. And I want to tell you a little bit of a story about early in my career. We just got an NIH grant, Dan Sonnenschein and Chuck Aberson and myself. And 3 months into that NIH grant, we realized our hypothesis was wrong. Ticks don't make JH. Paul's niece's work clearly was showing that. And I didn't know what to do. And just by total accident, the International Meeting of Juvenile Hormone was in Woods Hole. So all the people around the planet was at that meeting. Just totally by accident, I booked a hotel room in the same hotel as Larry Gilbert, who was a leading insect physiologist. He was department head at UNC in Chapel Hill, moved there from the University of Chicago. Just by accident, he was swimming one day when I wanted to go to lunch. A shortcut to the place where I wanted to have lunch at was through the pool area, and he saw me and he said, "Mike, how's it going?" And I said, "Larry, I don't know what to do. I got a brand new NIH grant. I proved our hypothesis is wrong. Ticks don't have JH." And Larry just said, made one statement to me. He said, "Michael, that's the best thing that could have happened to your group." It took me a few days to realize the significance of this, but for any of the young people that might be in this this presentation, remember it's the things that you don't expect that are the things that are the most exciting things to look at. And the fact that they didn't have JH, he was correct, was the best thing for us because it opened up so many other doors. And ticks don't have to be like insects. This may be also the case for how insects smell and taste, or how tick adults molt in a sense because they have these neural peptides that regulate molting, and this idea that ticks can actually see light, infrared light, by a particular mechanism that is not completely delineated in the American dog tick to detect hosts using their front pair of legs. In fact, you can see them using the binocular vision that they have with these two halos organs as they orientate and find an infrared light source. So this is the end of my presentation. Sorry for the glitches. I don't know what happened, but I think the loading of this laser pointer or the fact that we're recording, Emily, kind of delayed everything because everything worked fine yesterday, but I don't know.
I know, it's always like that.
Yeah.
It's fine. We have a couple of minutes for questions though, and so if people wants to have specific question, please feel free to chime in.
One thing I have to say, and probably Dan, I don't know if Chuck is here on this meeting, but one thing I have to say is that this work has created more questions than we had when we started by orders of magnitude. Because now we know so much more, but we know so much less because of what more we've uncovered. And that's always what makes research so much fun.
Mike, can I offer a question or comment?
Yeah, Dan.
And this is a warning to the more senior scientists, and that is be careful what you think you know, because Mike will come along and make you test it. And it's possible that what you thought you know isn't true. And that was the example he gave of when I said, "Why are you telling me to cut off their forelegs? I know that they need them to find the host and to blood feed. That's been established for decades. It's a waste of time. I know this." He said, "Do it anyway." And guess what? They did blood feed. And once I overcame my embarrassment, I had to recalibrate. You know, as the little lady says, when you're going in the wrong direction on the and you're trying to follow your GPS pass. Wow, how do I explain what I thought I know so well isn't true? Well, of course, there was an important moral here, and that is we are continually learning and reevaluating what we think we know, and there's always more to learn. So thanks for that, Mike.
Yeah. And who would have thought, Dan, when we first started this project with the Hallows organ, that we would end up with understanding that they're using probably this capsule area to detect light, to detect infrared light or heat, right?
Mm-hmm.
And that wasn't initially on our radar screen, and we stumbled upon that by accident. So we're continuing to have fun with this, and there's so much more to understand, like the role of that JH pathway in ticks, and what is the actual precursor to juvenile hormone that ticks are making. That pathway is not there just because they want to have this pathway. It's there because it has some kind of significant function, would be my guess.
We have a question from Anastasia. Have you looked closely into how repellent interact with Haller's organ, like DEET or picaridine? I don't know how to say that.
We haven't done any. I'm not sure I completely understand the question, but we haven't. The only experiments we did with the Hallows organ was the experiments where we cut off the end of the legs. We only focused on DEET because it's such a well-known repellent. And we know when you remove the Hallows organ and you put DEET on a surface, it does not repel the tick at all. It doesn't know the DEET is there at all. It walks on the surface as if it's not treated with DEET. So the evidence that we generated was that it's exclusively the Hallows organ that's detecting the presence of that repellent. We haven't looked at picaridin or other repellents that are available that work on ticks. That's the extent of what we did relative to repellency. And maybe the Mite people in the group here can. I mean, do they have Hallows organs? I think they do, don't they? They have something like the Hallows organ.
Not exactly, but they do have Sensilla on their forelegs. So we've studied this in Varroa. You know, the bee mite is partly responsible for colony collapse in bees.
Yeah.
And yeah, they have a cluster of Sensilla on their forelegs that resembles the Hallows organs of ticks, but it's not as well developed. It's not as consolidated, compact together. That's an avenue for new additional research if somebody wants to go down that pathway.
Okay.
Hey, Mike.
Hey, Saeed.
Hey, Mike.
Hey,
Good to see you. Hey, Dan.
You
Hello,
look the
Saeed. Good to see you again.
Thanks, Dan. I just have a comment. Did you look for the ionotropic receptors in this? Because it's a new class of receptors discovered in insect, the ionotropic.
Yeah, I think Anne found that. I'm not sure if it's in the paper that we published, but she did find a sequence that looked like an ionotropic receptor. Yes.
Yeah. Also for these gustatory receptors, they don't exist, especially on those test organs. And in insect, they exist on legs, on proboscis, on wings. They just exist everywhere. So they are not specific for the first leg of the ticks, I guess. Because of that, they exist everywhere on the insect body. So I guess they still exist everywhere on the tick's body also.
I know we found that transcript. I don't know if Anne's in here in this presentation, but I looked at her paper early this morning to remind me that whether it was in that paper or not, I didn't see a discussion of that. So I'm not sure if she actually published that discovery or not.
Yeah, we have a problem with understanding chemoreception. Briefly, the way it works in insects, as we all know, or should know, that odor and binding peptides play a crucial role. But then there are also these enzymes that degrade the binding of that ligand to the receptor. So it's a esterase that comes along and breaks that down. So after the initial depolarization of the dendrite, or the dendrite's ready to fire again because the ligand, in other words, the chemical that's been absorbed from the atmosphere, is no longer touching it. And then it repeats and repeats, and it's continued to fire repeatedly like that. Without odor and binding peptides, how do we explain how the ligand, which is a lipid, which, you know, it dissolves in the lipid coat of the CETA, and then get down through that pore. Okay, so now it's going from the exterior into a liquid, into an aqueous environment. And as we know, lipids and aqueous don't mix, you know, they separate.
Yeah.
So it's capturing that and transmitting it to the tiny receptors on the dendrites, which are right there. Somebody's mentioned the ionotropic receptors. Okay, that might be the receptor that's, when activated, triggers the depolarization event. But what allows the ligand, which is a lipid, to get there? And we're baffled. We just don't know.
Yeah, we had Pickett, Dan, look at our data sets, and he had some algorithms designed for finding odor and binding proteins, and he came up negative with the data set that we have.
Yeah, they're not there.
Yeah, but our transcriptomes were really, the contigs were really short, and that was really made the whole thing very challenging in trying to interpret this.
Absolutely. So this is a mystery, and another example of your beginning remarks, ticks aren't insects.
Yeah. Also, Dr. Khalil was a really integral part of all this work, as well as Dr. Thompson, because they were trained in molecular biology, and Dan and I, we were learning molecular biology.
That's right.
So a lot of the stuff that you were seeing was because of this coming together of people from different places with different backgrounds that allowed us to accomplish the things that we were accomplished. And that's why I wanted to make sure people understood that it was these young folks, these young researchers that came together that made all of this possible.
Thanks, Mike.
Yeah, it's good to see you, Sonny.
All
Been a
right.
long time, man.
I think we're gonna have to wrap up. So we are over the time right now, and there's still a couple of questions in the chat, but if you you can always email Dr. Rowe. I can put your email if that's okay with you in the chat if people have further question. It was about the control mode of action of insecticide. So I can just put your email in the chat, and I think I'm gonna thank you for your amazing presentation and for everyone to show up today. And please, if you're are interested in archaeology, stay tuned because we have a couple of more webinars coming this year. So again, thank you, Dr. Rowe.
Yep, I enjoyed it.
Enjoyed it, Mike.
See everybody.
Excellent presentation.
Bye.
Thanks, Mike.
Yep. Good to see you, Saeed.
Good to see you.
I got rid of your samples, by the way. I've been cleaning out freezers.
Yeah.
All the way from Egypt. That's wonderful. What a wonderful.
Yeah, what a wonderful thing.
Thanks, Dan.